All right, welcome back everybody. Today I will briefly go over using Snakemake to make our pipeline that we had already made for RNA sequencing, or that we had gone through for RNA sequencing analysis, a little bit more streamlined. So we'll be able to run this one line of code once we have our workflow built, and then we can apply that to other data sets. If you guys find this video interesting and you want me to finish the Snakemake video, let me know. As of right now, I'm not planning on doing the whole video just because it does take a long time. But I'll get started with it and kind of explain the different things that go on in the Snakemake file to do the whole workflow. So I keep saying workflow. What exactly is a workflow manager? So basically, last time when we had analyzed RNA sequencing, we had our raw read QC, we trimmed our reads. We looked at the trimmed, like the ones that were kept from there. We aligned, we did all that. So that's kind of our workflow or a pipeline. And Snakemake is a manager for that. And like I had previously mentioned, if we have our P53 data and now we want to look at another mouse data set that was RNA sequencing and we have our raw reads, what we'd be able to do is go ahead and just run this one Snakemake line, and it would go through and do all of the exact same things or whatever we put into the Snakefile on that data. So if we have a lot of RNA sequencing projects, we can make this one Snakemake file and just apply it to all of them for consistency's sake. So to get started, I made a Snakemake folder, and actually, yeah, so we'll make a directory called Snakemake, and then we'll go inside, so cd Snakemake, oops. Cannot create. Yes, I know, there we go. All right, so now we're in our Snakemake folder and it's empty. So what we have to do is we have to, here we can do this in Windows Explorer. What we can do is look at here and we can start adding our folders. So like with the last time we had our raw reads folder, we also have trimmed reads. And then if we want to run, for instance, QC, we can do raw QC, we can do trimmed QC, and then today I'm just going to do up to star aligned. In addition to these empty folders that we have, and then we're going to populate raw reads with obviously our raw reads, and we can go ahead and do that. On the GitHub page, there is a subsampled raw reads file or folder, and I'm just going to use those so it doesn't take so long to run. So I'm going to copy them actually. There we go. So now we have all of our SRR accession numbers in our raw reads folder. Okay. In addition to that, when we are doing everything, we have our for trimming, so raw QC, we don't really need anything extra. For trimming, we need that adapter file. So when we were looking before for our adapter file, that's in the mini conda folder in the home directory of Linux, and not on the Windows side, but you can find it in the Windows side if you really wanted to dig for it in the root fs for Ubuntu. Anyway, so we have our adapter file. So now we can do trimming, QC, and raw QC. In addition to that, we actually need our star index that we previously built. In Snakemake, you could make a step that creates the star index, but here, since it takes a while and we already have it built from last time, I'm just going to go ahead and do it again. So do star index. And if we go inside, I'm just going to copy over the pre-built index. You can see they're really big. The essay is a huge file, 20 gigabytes. But yeah, in the Snakemake, you'd be able to make a rule to build this out. So done a lot. Anyway, okay, while we're waiting for that, so we have our index file. We can also add like our GTF file. So our GTF is the annotation of the genome that came in that musculus folder. We can do just a folder called mouse. And in here, we can just add that, well, we can add both of them. We'll add our GFF, which is that annotation file, and it's not showing, oh, so here's the genome file, and then we have our annotation file. So if we wanted to go through all the way to feature counts, we'd have our GTF file to do so. All right, what else would we need in here? I think as of right now, that should be it on the folder structure side. Now we can move to building our environment. So the nice thing about having the environments, like I mentioned in one of the first videos, is that you can export it. Then you have this YML file, and then from that YML file, you are able to build a new environment, so to speak, that has the exact same programs and everything already installed. So if you're going to do Snakemake for RNA sequencing, you can make a environment that is specific for RNA sequencing, and then when you go to launch the Snakemake, you just activate this environment, and you don't have to worry about conflicts or anything like that. So what we're going to do is we're actually going to export the previously built tutorial environment that we had. So we built that and we ran our RNA sequencing with it. Now we can export it, and then we'll be able to rebuild an environment based on the exact same things from that. So in order to do that, we have to have the environment, or we have to be inside the environment. So if we do conda activate, then we can do tutorial environment. All right, so now we're inside of our environment. Let me make sure you can see. Yeah, so we're inside of our environment. Now an easy way to export this is going to be conda environment, env, and then export. And then we can point it to a file which we will then later be able to use again. So we'll just call this p53.yml. So it goes through and we'll open it up. In this case, I'm going to open it up and edit it a little bit, just because I want to rename the environment. If we were to go through and build p53yml again, what would happen is we would build tutorial environment, just because in the YML it's named that. So we can do conda deactivate. All right, now we're in base. So let's go over here and edit our YML. Normally you wouldn't have to do this. If you have this YML and you share it with somebody, then they can build the exact same thing and they'll have a tutorial environment too. So we'll edit with Notepad++. We'll get this over here. All right, so let's see, it's called tutorial environment. We can call this P53 workflow. We'll save it, and then we'll go back over and we can now build with it. All right, so we can do conda environment, we'll create, and then -f is going to let us specify our YML. So our P53 YML. So it's going to go through and it's going to install everything, add that environment to our list of environments, and then we'll be able to actually get inside of it, and we'll have all the exact same tools that we had inside tutorial environment. All right, cool. So now we have our P53 workflow environment built. An alternative way, if you're on the same machine and you're not going to actually be sharing this YML file with somebody, is you can do conda create name, and then you can specify In this case, it would be P53 workflow. And then clone, and then we would specify here tutorial environment. But I really like having this YML file because then you can upload it to a GitHub if somebody else is going to use it, or if you use it to make a publication with, like your analysis, then you can share that directly, and it has all of those. See if it'll open. Has all the versions and everything that is used in order to install a particular library. All right. So now that we have our environment, let's go ahead and activate it. So we can do conda activate P53 workflow. And right off the bat, what we're going to need to do is install Snakemake. So we can do conda install Snakemake. So that's another tool within Snakemake that can be shared with that YML file if it's, see if it'll open there now. Yeah, so if we go down, so you can see FastQC was in here, installed. So if we export a new P53 environment, we'll be good to go. So since Snakemake is built with Python, that means that we can use some Python syntax with it, like tabbing and importing different packages if we want to do things like reach out beyond specifying exactly the file name inside of the Snakemake file. We can search through a folder, which I'll show here, as well as indentations. So when we make our Snakefile, inside of that, we will have to indent kind of like the Python loops to designate whatever falls underneath of this belongs to a particular rule. All right, so since we're here, we can do in the home or like root folder of our project, we can do touch, and we'll do snakemake snakefile.py. So if we go over to our folder here, we have snakefile.py, and we can edit that in Notepad++. So it's really empty. We're going to start filling it in. The first thing that we have to do is import, or that we are going to do, you don't necessarily have to if you want to, like I said, specify exactly which files you're going to use. But we're going to reach out to our raw reads folder, and we're going to pick all of these out through the snake folder or snake file. So to do that, we'd have to import OS. We also have to import glob or globe. And what those are going to do is allow us to do that, reaching out into the folder and identifying things. And also what glob is going to do is it's going to allow us to throw some wildcards, so we can use what Snakemake is calling wildcard. You specify a variable and it'll automatically detect. So in this case, we will, here, let's just show this first one. Okay. So the first one we're going to call our files into Snakemake. And to do that, let's try to break down what we're actually looking at here. So we have our SRR number or SRA number, and then we also have this 1 and this 2 for each one. So each file can be broken up into two different pieces, the SRA number as well as like forward and reverse. So when we go into Snakefile, we can do something like SRR or SRA to pull our SRA number. And then we can do something like forward, reverse, read. So then we're going to have two different variables come out of this. One is going to be the SRA number, which is going to be the SRR 2121, whatever. And then we're going to have an FRR, which is going to be either a 1 or a 2. And we can do glob underscore the wildcards. And then in here, we want to point our Snakefile to whatever folder we're going to be working with. So this is going to be raw reads. And then we have to split this up into SRA number as well as our forward reverse read. And then .fastq, right? All right, so then in here, we can call this one lowercase SRA and this one lowercase FRR, forward reverse read. Now when we run our Snakefile, it's gonna automatically detect all of our SRA numbers, and it's gonna detect 1 and 2 for our forward and reverse reads. All right, so now what always comes at the top, and it doesn't have to, is gonna be rule all. So what this is gonna be is a rule that is terminal. So with things like trimming, you have your raw reads coming in, and then you have trimmed reads coming out. But your output is then going to be used as an input for mapping to the genome with star, right? But with things like FastQC, the output from FastQC doesn't necessarily have to go into another rule. And because there is no rule that's going to be pulling the output from FastQC, that would go here in rule all, if that makes sense. So any rule that you make that has an output that can go into another output, an output that can go into another input of a different rule doesn't have to belong in rule all. But any rule that does, like it's the last step. So if we were going to run through all the way to feature counts would have to go in rule all because there's nothing else to pull that from or pull that to. So input, we would designate what has to come into rule all from another one, but we're going to leave this empty just for now. So the first thing that we want to do is run FastQC. So we can do raw FastQC as a rule. All right, so then for our rules, we'll have things like input, like with rule all. We'll also have our output. And in the case of this QC, we're gonna have to put our output here into the input of rule all, otherwise it won't run the raw FastQC. All right, we'll have something like threads, so we can specify how many threads this rule uses, and then we'll have this shell. So what actually is this rule passing to the terminal to run? That would belong here in the shell. And I always run mine with 3 quotes, but you can do one at the beginning and one at the end too, and it would work just the same. So we have a raw FastQC, and then we would have something like Trimmomatic. Hi. All right, and then we could have another rule for star. So rule star. Okay, so for our raw FastQC, the input is going to be our raw reads. So you can just make a variable here called raw reads. We can call it raw read because it's going to be a single read that comes in here. Equals, so what does this equal? This is going to equal something from our raw reads folder. And then we're going to have to specify a particular ID. So we have our SRA number. And then we're going to have to specify a particular forward reverse 1 to something, and then the extension FastQ. So here we can do lowercase SRA, and then here we can do lowercase FRR. And then what is the output from FastQC? Well, we have a zip file and we have a HTML file. So the zip file contains the information, like the raw information, and the HTML is like that beautiful page. So then we can make 2 variables. We'll call one zip equals, and then HTML equals. And we would only actually have to do probably one of these in the input up here, but we'll just do both of them here for the sake of being complete. So we're going to put this in our folder. We made one called RawQC. We'll point it to RawQC slash, and then we want the same SRA number in here, underscore, and then FRR, and then underscore. FastQC is automatically going to add this last tag on called FastQC, and then .zip in this case, and then we have to put a comma at the end because we have another variable, and then we can do the same thing here and just rename this HTML. All right. So now we have our input for raw FastQC. It's going to be our raw reads and then a particular SRA with a particular ID, 1 or 2, forward, reverse. And then the output is going to have that same SRA number and the same ID or the same forward, reverse designation. So that's what we have to now tell all to look for. That way our Snakefile will know that that job was done and it can begin running another one. So here we can do expand, and then we'll just call RawQC again, slash, and then in here SRA, underscore, bracket again, FRR, underscore, FastQC, dot, and then we can add another wildcard. We can call it extension, for example. And then now we have to specify at the very end of that what exactly each one of these are. Okay, so SRA is going to equal a capital SRA that we set up here. Our FRR is going to equal a capital FRR that we set at the top again. And then extension is going to equal one of two things. It's going to either be zip or it's going to be HTML, just like down here. So we have basically this. If we expand this out using expand, we will say, okay, for all the SRA numbers and all of the forward, reverse reads, and for both of zip and HTML. That's basically what this is saying. We have to look for that when it's done. Threads. We know that FastQC is single threaded, so we can put a 1 here, and what that's going to do is it's going to tell Snakemake, okay, if you're going to give me 8 cores, we're going to run 8 raw FastQC files or FastQC jobs at one time. And then in the shell, just like if we were going to type out in the shell, we do FastQC, or in the terminal, we do FastQC, and then now we can start specifying some of these variables that we set here. So we're going to feed in our input and then dot raw read. And then we'll do threads, and then, so it's not square brackets, it's kind of the curly ones. And then we can do threads here. So whenever we put something in these curly brackets, it's going to look up here for the name, whatever you put inside. So we have raw read, it's going to look for raw read. And we have input.rawRead, so input raw read. All right, and then dash output. So we need to add one more thing in here. We're going to add a parameters list, or variable. We're going to just call this path, and it's going to be equal to RawQC. And the reason that we are doing this is because we can't specify with FastQC our zip and our HTML, for instance. We just specify a path and it'll automatically put our finished file in there. That's with a lot of these tools. So we have to create a parameter, which is just like a miscellaneous ID or a miscellaneous variable here. And then we can put that down here. So we can do it param, needs an S. params.path. So now it's going to look in the params for a variable called path, and then it's going to spit that out. All right. So now we have our FastQC done. We can go ahead and do another one for Trimmomatic. So the input for Trimmomatic is going to be our raw reads again. So we can do, but we need two, remember. So we have to do forward. So we can do read one equals, and then read two equals. So read one is going to equal our raw reads, and then we'll just specify an ID this time, and we're going to have to say read one, comma. And we have to do this because if we were to put something like this back in, it's only going to feed in one read at a time. And if we want to pair end read, we have to do both, which we have to designate 1 or 2. So we'll do raw reads / brackets 2 this time, FastQ. So I'm back in here, we do IDs and IDs. The output from Trimmomatic, we're going to have, we can just do 2. We'll actually get 4 outputs. Remember, so we have our paired, forward and reverse paired, and we have our forward and reverse unpaired. So like forward paired, forward unpaired, reverse paired, reverse unpaired. But here we only want, we could do forward paired equals, and that's going to end up in our trimmed reads folder, and we'll specify an ID, and we want 1p, we want to look for 1p, and then .fastq. Okay, and we also have reverse paired equals trimmed reads / what will be an ID, and then 2p .fastq. So we go back in here and put our IDs. So we want the same name coming to the output as what's in the input, which is pretty logical. And then here we can do 4 threads, for instance. And since we're only using the small reads, it's going to go really quick. All right. Now we can work on the shell. Just like before, we'll do Trimmomatic. Do Trimmomatic, and then we want paired end, like I was saying. Threads, we can specify, look at our threads variable. Okay, and then just like with Trimmomatic, we can specify our input reads now. So we have input dot, we want read 1, and then we also need input dot read 2. Okay, so instead of specifying our exact outputs, what we can do is specify, again, another parameter with the base name of the output. So the base name will have just our ID with FastQ, and then what it's going to do is Trimmomatic will automatically add in the SRA number. So we got to add this SRA. It'll automatically add in the SRA number, and then 1p or 2p, as well as 1u and 2u. Okay, so we'll do base out. We just want 1 dash. So we need that parameter variable. So params. Okay, we can do base name equals trimmed reads / SRA, and then .fastq. Always with the S. All right. And then in addition, we'll do a log file. That way, if we wanted to, we could do multi-QC and get all the information about our Trimmomatic run or runs, which would be in trimmed reads / SRA, and then .log. Pretty easy. Okay, let's make this a little wider. we have our base out, and now we need to do params.baseName. Okay. Next, we'll do the Illumina clip. Illumina clip, and then TrueSeq. So we're going to add in our adapter names here. 2, 30, 10, 2, and then keep both reads. I don't know why it won't come back. There we go. Just save it. And then leading, we'll do 3. Trailing, we'll do 3 again. And then min length, we'll just set that to 36 as before. And then we will spit it out to, with the 2, it's going to look for the output. And then we're going to point it into a file, which will be our params.log. All right. So, good deal. Next, we can do our star. So the input from star is going to be the output from Trimmomatic. In order to do that, we can do like read 1 again, and then we want to set it to equal to our rules. So we have all of our rules. We're going to look through our rules for a rule called Trimmomatic. And then in Trimmomatic, we want to look for our input, or we want to look for our output, my bad. And then we want to find our forward paired, and that's going to be equal to read 1. So every single time we don't have to go back through and specify the path to each file. We can just specify the output from Trimmomatic. We want to look at that now as the input for star, comma, read 2 is going to equal just the other one. So rules Trimmomatic dot output dot reverse paired. All right. Now the output from star, in this case, as the shorter video goes, we're just going to look at 2 different things. We're going to look at the BAM and the logs, but you can go through, like when we ran star before, we had the mate 1 and the mate 2, which were unmapped reads. We had actually 2 different log files, but we're only going to look at one of them. You have the BAM, a lot of stuff. But we'll just look at the BAM and a log file. We won't look at both of them. So something that I noticed going through, I will often forget the comma at the end, just because usually when coding in like R or something like that, if it's a single line, like if you're setting a variable, you don't have to put a comma at the end. So it's just something to get used to, but yeah, don't forget the comma. So the BAM is going to be equal to our star aligned. So we have our star aligned folder. We're going to look in there for a file called whatever our SRA number is, and then aligned dot sorted by chord. So we're going to specify in star that we want to sort our BAM file by coordinates, .out.bam. All right. Hi. Then our log file is going to be, what was it? Star aligned, that same folder. And then we want our ID again, so SRA, and then log.final.out. So those are going to be our 2 outputs that we're going to look for. And then for star to run, we'll have to put that up here. But since Trimmomatic is being called in star, so we're looking for these from Trimmomatic, we don't need to specify Trimmomatic up here in rule all, just the terminal ones. The number of threads, since I don't have enough RAM to run multiple loaded genomes at a time, we'll just set like 45, and then it'll run one at a time if I give it my CPU. What else? Let's just work on our shell now. All right. So for our shell, we will need to call star, and then run thread n, and then we can give it our threads. Genome directory, and that's what we copied over from our previously made index. So this is going to be our genome directory, star index, and then like before, since we're going to run multiple mappings at one time, well, we don't want to load our genome and then unload it every single time, because loading the genome takes a decent amount of time in itself. We will do genome load, and then we can do load and keep. So the first time what it's going to do is it's going to go through and it'll run the step to load the genome into memory, and then after that it'll keep it in memory. The only thing that we have to do is at the very end remember to do the remove. So I might actually have one in memory right now. So if we do star genome dir and then star index, and then genome load remove. Oh, I need another dash. Okay, so we have our genome removed. What we can do now with our genome load keep, or after that, we have to read our files in. So we do read files in, and then we want to specify our input, read one, which is going to be our Trimmomatic output, and then we have our input read.read2. Okay. Now we'll do just like last time, out filter intron motifs. Yep, make sure I spelled it right. And then remove non-canonical reads. So you only want the ones that are going to be the typical splicing for RNA sequencing. And we have out file name. And then that's going to be our params. So in parameters, we have our prefix, which is going to be star index, and then the SRA that we have from there. So params dot prefix. Good deal. Now out SAM type. So Snakemake is going to look for a BAM file. We're telling it to look for a BAM file. So for the out SAM type, we need to specify BAM, and then we want it sorted by coordinate. So we tell it that we're going to have the output named sorted by coordinate. And then since we're doing the genome load and keep, what we have to do is we have to tell it to remove, or no, we have to limit the sorting RAM allocation. Otherwise, it's going to yell at us for that too. So we'll limit BAM sort RAM, and we'll just do 5 gigabytes, so 5 billion, 123, 223, 323. And then we'll keep the unmapped reads again. So out reads unmapped, and then it'll be fastX format. All right, so let's run through what we have so far. So we have our imports. So this is just typical Python. We're going to import OS and glob or globe, however you want to pronounce it. We're going to look through our raw reads folder for SRA numbers and the forward reverse designation, forward reverse read, 1 or 2. From there, we're going to feed it into our rules. So Snakemake works backwards. if you can kind of think of it that way. So what rule all is going to do is it's going to be the last thing that's actually called. And so if we look at, so it's actually the first thing that's called, which then works backwards. So raw QC is going to be the terminal job here. And so we have SRA and FRR that we need to look for, and we're defining those variables here. So in this rule all, we're telling it these SRA numbers equal the SRA numbers from the files, and the forward reverse read is going to equal the forward reverse read from the file, and then the extension is zip in HTML. What that is doing is it's going to tell rule FastQC that this SRA here in the output equals the SRA in rule all, and then consequently the SRA number in the input equals the SRA number in the output for FastQC. This star aligned I typed out is telling the same thing for star. So the output from star, SRA number and then a line sorted by coordinate out bam, that SRA number is going to equal the SRA number from our input files. So if we go to rule star, the output has these SRA numbers. And then so the input, which is the output from Trimmomatic, is going to have the same SRA number, follow it all the way through. And then just like the FastQC, the input for Trimmomatic will have the same output or same SRA number as the output for Trimmomatic. So it kind of follows itself all the way through to the very end, which is why we need rule all called for those terminal jobs. That way we can say these wildcards that are being called, that's what they actually equal. So we can go ahead and give me 1 second here. I ran this as a test just to make sure that it worked. Okay, we should be good to go. So let's talk a little bit about what some of these, I need H. Yeah, it'll work. So what some of these flags are. So we have dash, we can do dash dash C or dash dash cores and give it the number of cores, or we can do dash J and give it the number of cores. So we do snakemake dash H, and we'll scroll all the way up to the top. We can see we have, or we can do a dry run. So a dry run is going to be dash N, and we'll do that first just to make sure that our Snakemake is structured correctly. We have to make sure that our rule all is calling the right jobs and creating the right files. We have our cores. So our cores are going to be dash J. So we look down here, we have our cores, which are dash J. And then another thing, since we have our snakefile.py, I call it .py, that way in here we'd get the colors for specific things. But since we have that extension on there, Snakemake won't automatically recognize it. So we have to do dash S and specify snakefile.py. If you were to remove the .py, then Snakemake would automatically pick up the snake file. But in Notepad++, it just makes it a little bit nicer as far as like keeping tabs and everything. All right. So to make sure that it all works well, what we can do is Snakemake. We have to specify dash S and then snakefile and then dash N. So dash N is going to be that dry run. Just make sure that it works. And it looks like we have our structure set up right. The only thing that this dry run won't do is tell us whether or not we have an error in our shell. So there's a very real chance that there's something wrong here in our shell commands that we won't know until we do it for real. Okay, so we have one count of rule all. So rule all only gets called one time. we have 24 raw FastQC. So if we look at our raw reads, we can see that we have 24 files, which is the same as before. For what would come next is Trimmomatic, and Trimmomatic has 12. So we have 24 reads, we have 12 forward and 12 reverse, and then star is going to use the output from Trimmomatic as its input. So that's also going to be 12 for a total of 49 different jobs that Snakemake is going to launch. So now that we did that, what we can do is try it for real. -J, and then we can do 64 cores and see if it works. So all the green is good. And then we are mapping already. So the mapping part will take a while, but yeah, it looks like everything worked out like it was supposed to. And then you can always go through and you can do like a rule QC for Trimmomatic. You can do multi-QC in here. Like I mentioned at the beginning, you can do a rule to do the star index. You can do things like a config file, and in that config file, you can specify particular threads. You can specify your input files. Somebody else in university had it specify a particular genome file or like a genome file location, and the GFF or the GTF annotation file location, all those types of things. And then in here, you would just call that shorter. So like here, you would call that as a variable, kind of like the threads. You'd call the index or what have you as another variable. All right, we can look through some of these folders. So we did raw QC. So our raw QC folder will have all of our QC in there. Trimmed reads, here we go. We have all of our trimmed reads in, as well as the log files. So open Notepad, we have our adapters, and the information about the surviving for forward, reverse, paired and unpaired, as well as our align files. So if you go in here with our log final out, so this is what Snakemake is actually looking for this, as well as the BAM file. Oops. What we can do is inspect this guy and make sure that we're getting good results. So there we go. We have 82% uniquely mapped reads for this file. And so these are all subsampled files. That's really small anyway, but so yeah. And then Snakemake is done. So now all of our star file or our star mappings are done for all 12 samples. So yeah, if you found these helpful, please like, subscribe. If you have questions or you want me to like do the entire pipeline with Snakemake, feel free to let me know. I think it'd be really, I like doing the Snakemake stuff. Sometimes it can be troublesome if it doesn't work out like you wanted it to, but yeah, thanks for watching, guys.